Hey, hi, hello everyone. What are you doing? Enjoying your life? Oh, get prepared. Your examinations are getting started soon. So just don't waste your time and sit with pen and paper and start preparing now. You know what? Today I have a very important promises to be done. What are the promises I'm going to make? First promise, this chapter, this video will be a life-changing video for you. That means if you watch this video completely, few of the scores you will secure in your bank, right? Second promise I'm going to do that this will teach you to draw the diagram. Means once you will watch this video, will draw with me, you don't have to do it again and again, and this is no more going to be a very tough job for you, because I'll draw the diagram step by step in a very easy manner, so that it will be easy for you, because I'll break the diagrams and will make you understand about it. A very compact lecture today I'm going to give, so that you can understand it in a very less time and also with full and crystal clear clarity. So don't skip this video, watch the video till the end, because if you'll not watch the video till the end and will skip the video in between, this will be a very difficult thing for you to understand the whole concept, because from here short marks will be taken by you. So this is a very important topic, examiner's favorite topic to be asked every year. So watch till the end, and you know, if you like the video, what do you have to do? You have to hit the like button, share it with your friends. So let's start our amazing class. Okay students, so let's see what are we going to discuss in today's class. So first of all, we have to understand what is transpiration. That is the introduction of transpiration. Okay, first we'll understand about the transpiration, then we'll understand the demonstration of transpiration, like how can we see that transpiration really is taking place. Okay, how can we see that? Then measurement of the transpiration, that if the transpiration is taking place, how much water is getting lost, or what is the rate of transpiration, that we are going to measure in this topic. And then we'll understand the kinds of transpiration. Yes, transpiration can be done there are types of transpiration. It's not only one type of transpiration. So we are going to discuss that also. Factors which affects transpiration, that means those factors which increases or decreases the rate of transpiration. And again, guttation and bleeding, one of the very important topics from this chapter, that is guttation and bleeding. So I'll make sure so that you can understand this topic and understand the differences between guttation and transpiration. So those things also we will discuss in this chapter. Again, at the last we'll do significance of transpiration. Significance means importance, or why is transpiration taking place, how is it beneficial. So that we are going to discuss in this topic. So let's start. First of all, we'll start with what is transpiration. So we all know that transpiration is nothing but a kind of loss, right? Which kind of loss? So transpiration is the evaporative loss of water from the aerial parts of a plant. Transpiration is the evaporative loss of water. So actually it is a loss of water. Then why transpiration is important? See, transpiration is very important for the cooling effect, that we are going to discuss in the significance of transpiration. So first help which transpiration gives, and that is providing cooling effect, not only for plants but also for ours, for environment, right? One of the very beneficial act is this. Second one is it also provides the suction force, and I'll be discussing about suction force and everything in meanwhile. But let's understand these are the two benefits, that is why transpiration is taking place. So actually transpiration is the evaporative loss. So evaporative loss means here not water droplets but the water vapor goes out, right? Water vapor means water is getting lost, or loss of water is taking place in the form of water vapor, right? Water vapor is going out. Now the second thing is from the aerial parts. What are the aerial parts?
Aerial parts are the stem and the leaves. Stem and leaves. Stem and leaves of a plant gives the we can say the transpiration taking place from the aerial parts, and these are the aerial parts. So I hope you are now getting to know why transpiration is very important and what is transpiration. So this we understood, transpiration is the loss, evaporative loss of the transpiration. So we all know that, see what happens, that the plant absorbs water from the soil. Okay, plant absorbs water from the soil. Only 2% of this water is being used in activities done by the plants. Rest of it goes out through transpiration. So I hope this is very clear that plant, whatever plant are there, they absorb waters from the soil. Whatever water they absorb of these water means of the amount of the water, 2% only is being used for the activities they perform, like photosynthesis and all. So rest of the water, where does it go? It goes out from the plant through the process called transpiration. So that means this is one of the very important process, and like transpiration takes place in a we can say major scale. Okay, this is taking place in a major scale. So that's we understood. Now let's discuss the next topic, which is the experiment through which we can demonstrate actually that transpiration is taking place. Let's understand the first experiment. We have 2-3 experiments, basically 2 experiments we have majorly, and the question comes from it many times. So just be very careful while studying these experiments. See, first experiment tells, here we have to take let's take a well-watered pot with a plant. Okay, well-watered pot with a plant. Tie the plant with a polythene bag. Okay, now let's understand one thing here. When we tie, you can see we have tied till the stem. Okay, we have not covered the whole pot. You can see we have not covered the whole pot. This pot is as it is. Why is this so? Question has been asked, why is this so? Because we have to let the soil evaporate. Okay, let the soil evaporate. Okay, that we don't want this droplets to be seen due to the evaporation of the soil. We only want to see the transpiration, not the evaporation of the soil. So soil is also obviously watered, so it may also evaporate. That is why we are not tying the polythene bag to the pot. Only the stem, till the stem we are tying, and tightly tie it, and keep this pot in the sunlight or under the sunlight. This is under the sunlight for 2 to 3 hours. Okay, or approximately 2 to 3 hours will work. So we have to keep this setup to the sunlight. Okay, and after 2 to 3 hours we can see the tiny droplets over the surface of the polythene bag. Okay, you see this surface, these tiny droplets. These tiny droplets are nothing but sign that transpiration has taken place. Okay, transpiration has taken place. Okay, now one thing more we need to understand, and so that I am writing it as a note. So note, tiny droplets will be seen only in the humidity or in humid weather. Okay, if the weather is humid, then only we will be able to see the tiny droplets. You know why? Because we know that evaporation means what? Once the water gets heated up, then it becomes vapor, and when the vapor, okay, that vapor comes in the touch with the cold surface, then it condenses, and once it condenses, it again becomes the water. So the process is, what is the process? Water becomes water vapor when it is heated. Okay, that means when the temperature is high. Now water vapor becomes water droplets again, water droplets, due to a process called condensation. And condensation will be taken place only and only when there is moisture in the air, and that is only called the humidity. Okay, that is only the humidity. If there is a humidity in the environment, then only we can see this tiny droplets. Otherwise, looking at this tiny droplets is very difficult. Okay, so that means water vapor can't be seen. Okay, because it's a vapor, we can't see this, but water droplets can be seen. So in this case only. So this is the first experiment through which we can actually check that transpiration has taken place. Let's do the second experiment. Second experiment has the same setup, almost same setup, but only here we have just tied the whole pot. You see, the whole pot is covered. We have to take a covered potted plant, and then this is a bell jar. This is a bell jar. So first we need to take a potted plant which is fully covered. Okay, and then we have to obviously make them transpire and keep a what? Bell jar on it. Bell jar provides a cool surface. Okay, bell jar provide a cool surface for the vapor to be condensed, so that the tiny droplets are visible to us. Okay, see, the same process we have done. This covered potted plant we have taken, and we have this bell jar on it, over it, and kept it for in the sunlight for 2 to 3 hours again, and then we'll see again the same thing. And here this tiny droplets is visible significantly, because we know that the bell jar is a cool surface. It provides the cool surface, and once the vapor collides with this surface, it just condenses immediately and makes the water droplet. And so it is very easy for us to see these water droplets and identify the transpiration has taken place. But there are two things, okay, which we call a control setup. So basically, in the first experiment also we needed one control setup. What is a control setup? I think, okay, what is a control setup? I think in class 9 only you understood, you must have understood that control setup is one of the very important part of any experiment. So that if you are doing this experiment and anybody says that maybe this is something else, this is not transpiration done by the plant only, maybe this polythene bag also contains some of the fats, globules, or maybe that is why this droplets can be seen. So for that we need a control setup, and in this control setup what will we do? We'll take the polythene bag and will tie without any plant. Okay, inside it there is no plant, and just we have taken the polythene bag and tied it. Okay, and kept it in the under the sunlight for 2 to 3 hours. Once after that, we'll see that there are no tiny droplets, such droplets on this polythene bag. That means this tiny droplets is due to the plant, not due to the polythene bag. Okay, so this is the control setup we needed to do here. And now same control setup will be here also. What will be the control setup? This control setup is here very interesting, and let's see what. So in this bell jar, in the first setup, let us suppose setup number A. In this first setup, what will we do? We'll just make in this way. We'll tie it and we'll keep a plant on it. Okay, and then this is the bell jar, setup number A. Okay, and then in setup number B, what will we do? We'll again take a potted plant. Okay, and then there will be a paper. There will be a paper over it. And how will we tie this paper? We'll either take the crop, we'll pin it, or we will take a cork slide, and on which we will keep this. So this is our setup number B. Okay, let's see the third setup, and which is setup number 3. Okay, in this there is no plant, just the paper. Okay, just this paper. Okay, there is no such plants, but only the paper. Now what will we see? And this is the setup number C. Okay, now what will we observe? Our observation is plant A, we will get the tiny droplets. Okay, in plant A, we will see this tiny droplets over the jar, on the surface of the jar, we'll see. Okay, so we can see that when we the same thing happens with the jar 2, jar B, but as we can see the tiny droplets are there, this paper turns pink. Okay, let's make for now red, but this is pink actually. Okay, this is pink actually. It will turn pink. Okay, now when we have seen the third setup, in third setup no changes, means this paper has no changes in its color. Okay, so what is the observation? A tells that the transpiration has taken place. In B, this paper is called chromium chloride paper. Chromium chloride paper, and its characteristic is it is blue when it is dry, and it is pink when it is wet. Okay, and also wet means when it is wet through water. Okay, so you see the chromium paper, this chromium paper has turned pink. Why? Because of the presence of water here, right? So that means it indicates that exactly water has been released by the plant, first of all we have understood that it was water. The thing which has released here is water. This is confirmed. Now second thing is it is been released by the plants only. There is no any like this is not the characteristic of a bell jar or a paper like they are releasing something. No, because when we have kept the same setup, and this is also called the control setup, right? Control setup. Okay, this is a control setup, and this control setup tells that if you are keeping the same setup, everything will be the same, only the plants is not there, plant is absent, then the same thing will not happen. This tiny droplets will not be seen, and obviously the color of paper will also not get changed. Okay, that means this is the whole thing
which actually tells and demonstrate that transpiration has taken place by the plant. I hope this is very clear to you. Okay, there was 2 experiments which we have done to understand that transpiration has actually taken place. Okay, next thing is about the measurement. So next topic we are going to understand how to measure the rate of transpiration. Although rate of transpiration means the amount of transpiration or the water being transpired. Okay, first method, see, these all are weighing method. 2 methods are there, which is weighing method, but first method is weigh the potted plant before and after keeping it under the sunlight for 2 to 3 hours. Okay, so what is the first method? First method is very simple and easy type of method, and this is not that important although. So what we have to do, this is the easy process. Take a small potted plant, weigh it, means find the like obviously weight of it, and then keep it on in the sunlight. Under the sunlight it will be kept for 2 to 3 hours, and then there will be transpiration taken place, and after that again weigh it. When you will weigh it, you will find the less weight now, because the loss of weight here has taken place due to transpiration. But not only for that, because of the transpiration, because transpiration plus photosynthesis plus the evaporation, everything has been included in it only. Okay, but we know that for other activities only 2% water is been lost. So obviously rest of the thing is for the transpiration. Transpiration has the major role in it. In the transpiration plays major role in the loss of water. So basically the whatever like loss amount will get majorly it is of transpiration. So only 2%, 1% will leave here and there. Rest everything is transpiration. So actually we are not going to get the exact amount of transpiration has taken place, but almost we can find out, we can measure. Okay, this is the first one, right? Now let's see the second one. So in the second one you can see the setup here. What happens here, let me make you understand this. In this setup what will we do? We'll see that water is going, this is the setup in such a way that when the water, this layer will decrease, okay, then water will come from this only. So this layer will also start decreasing. Okay, and this is also scaled, means it has a scale. So it is scale. Okay, so we can easily identify the amount less, means water decreased here, so we can easily measure that. Okay, what happens, and also there is a oil layer on the tops to avoid the evaporation, okay. We have taken a twig with roots over it. Okay, it has a roots. Now what happened when the plant, such plant is there, let's see how does it work. If you have a plant, okay, now this plant has root, and this is under the jar which is filled with water. Okay, which is filled with water. What happened here is the water droplets which will be transpired. Okay, once the transpiration will be done, we know that transpiration is nothing but the loss of water vapor. So it will go out, and here will be created a void. Void means empty space, right? Empty space will be created. Once this empty space is there, again other like water droplets will try to fill this empty space, and then in this process this place will be emptied. Again the same process will go on, and this void will be created. At the last this void will be created here, and once it is created in the root, then root, what will they do? The water outside the root will take place of it, and then again the same thing will happen, and it will again start taking the water from this area. Okay, and once this process will take place more, then this water layer will start decreasing.
and we'll get the water level, let us suppose till here. Okay, this water level has gone and we have the water level now from this area. Okay, so this is the water level. So from here to here, this is our scale, and we can easily measure how much water has been lost through this process, okay, or in this process. This is the way, this is the setup through which we can do the whole process, right? This is the second one. Okay, third one is the very simple one. Let's take a test tube. Okay, one test tube we will take, and in this test tube what will we do? We'll first of all we'll keep the layer of oil, okay, to avoid the evaporation. Question has been asked that why are we keeping the oil layer here? Because to avoid the evaporation. Then we have water on it. Okay, we have water. We have water in it. And now what will we do? We will take a twig. Okay, of course, why do we take twig in these situations? Because this is a very small test tube. We can't actually fill or actually keep the whole plant on it. Okay, so we need to like work with a twig here. Now we have a twig. Okay, let us suppose like this. Now the transpiration will take place, and once the transpiration has taken place, now let's see the same thing. This is A before transpiration, and now this is B after transpiration. What will happen after transpiration? After transpiration, this layer of oil will come to this way. It will come here, because see, a layer of oil is not getting evaporated because it's oil, and also it's not letting the rest of the water evaporated. So we can easily find a bar or a layer to measure this. You can see this, and now again the twig is already there. Twig is still there. Okay, so you see the setup B, setup A and setup B. You can see the layer of oil getting decreased. So this is what we can also do for the third method. This is our third method through which we can find out or we can measure the transpiration. Okay, again what will we do? We'll measure this amount. It was here and now it is here. So this is what we have to measure. If we can measure this amount, we can easily find out how much water is been lost through the transpiration. And again I am saying, when the transpiration takes place through these methods, we have to make sure that it should not go with the evaporation. Although it can also keep some of the water for their physical activities, whatever activities they perform, but most of the amount of water they use or they absorb and lost through transpiration only. So this way we can easily find out the way to measure the transpirational water or transpirational loss. Okay, let's see the last method for this measurement. Last method, that means the fourth method, and this is very important method. This method is called Gannon's. This method is called Gannon's spotometer. Okay, this method is called Gannon's spotometer. Why Gannon's spotometer? Because Gannon has given this. Okay, Gannon has given this experiment or this setup. That is why it is called Gannon's spotometer. Although this setup is called a potometer. Okay, so what are the things in this potometer? Let's understand. You see the air bubble. The major role is of air bubble here. Sorry. Let's see what are we going to do. This air bubble, what will happen? You can see this air bubble is now here, but it should start from the beginning. That is, this is the scale through where it has to start. Okay, it has to start from this way. So what happens? Let us suppose our bubble is not here. Okay, let us suppose this bubble is not at the initial point. Okay, but it is behind the initial point, like it is here. Let us suppose this bubble is here, not at the initial point. Okay, what will we do in this case? What we have to do is we need to take this, you see this capillary tube. Why capillary tube? Because this is a very thin tube. Although for purpose of like making you understand, we have kept a little broad tube, but it is more less than this, more narrower than this, okay. That is why its name is capillary tube. So you see, what we are going to do is, we are going to dip the tube again and again, so that the water gets in and start putting a pressure on this bubble, so that this bubble starts moving forward and coming to this place, okay. Our goal is to make this bubble bring to here, okay, bring to the beginning point or the starting point. This is our motto. So this is the condition when the bubble was behind the starting point. Let us suppose what happens when the bubble is ago, like this, okay. If the air bubble is before the starting point or ahead of the starting point, what will we do in that case? What are we going to do is, now the reservoir will be used, okay. Now the reservoir will be used. We will just loosen this spincher, okay. We'll loosen this spincher. Once the spincher, now it is very tighten enough, okay. What will we do? We'll loose it. When we'll make it loose, then this water will start getting pass, okay. When this water will start passing, this will force, this will push this bubble to the initial point, okay. It will push the bubble to the initial point, okay. This is what we need to do. So this is the whole process. Now how will we measure that how much water is been used due to transpiration, okay. See, when the we have taken a cut shoot means a twig without a root, okay, a twig without a root. So you see, what will we do? We'll keep a twig, and once the transpiration will be there, that means the water will be lost. Water loss will take place. This water start coming to this place, okay, coming to this place. Initially my bubble was here, but since the water started coming to this place, so the bubble will also get pushed and it will come to the final place like this, okay. It will come to the final place like this. So what will we do? We can easily find out the difference between this and this place and this scale, okay, this scale. When we will easily find the difference between this scale, we can say this much amount of water has been transpired or been lost through the process transpiration, okay. So this gives you the exact measurement of the water lost, but there are some limitations, okay. Limitations of photometer. So Gannon's photometer has some limitation. First of all, keeping the bubble on its real or original position is very difficult, very difficult. Second is, twig doesn't live for a long period of time, okay. I hope it is clear. First limitation is, you see this bubble, I have told you two methods. Once the bubble is behind, we have to dip the tube again and again so that it come to the original position. If we are ahead to the normal position or original position, we need to lose this reservoir and make it on its position. But this thing, as I am saying, then it seems easy, but it is not easy at all. It is very difficult, and we need to work hard for keeping this bubble on the right position, and this takes time also. During this time, when we are doing this, our twig, this twig is this dies, okay. This twig dies, and when it is dead, then we can't have any transpiration done, and when the transpiration will not done, this bubble will also be not checked, and the whole process will be wasted or lost. This process will not work at all. This process will work only and only when this twig is alive and having the transpiration done, okay. So that is why this photometer has some limitations. We need to understand that, okay. So limitations also used to come in your examination. So maybe that can be one of the very important
thing to be remembered. So limitation also you need to understand. So I hope this is clear to you. Now let's understand the next topic. So next topic is about the types of transpiration. See, let's understand first of all the structure. One structure I am going to tell you. See, I am going to tell you the sectional view or cross section of a leaf. Okay, cross section of a leaf. So this we are going to understand. So first of all, what will we make? We'll make the outer part, which is called the epidermal part. Okay, now we up to this epidermal part there is some waxy layer. Over it, some waxy layer is there over it. See, this diagram is very important, so just look very carefully to this diagram and try to draw it with me. Okay, so if you'll draw with me, it will be very easy for you. Otherwise, if you'll sit separately and try to draw, maybe it can be a difficult task. Okay, now what will we do? We'll see. We are going to make this layer, and this layer is actually very compact layer. Okay, column way. You see, this is column in columns, and they don't have any intercellular space in between. You see, they are tightly packed with each other. Okay, so this is the first layer. Okay, inside the epidermal also there are two layers which are called mesophils, and in this mesophils, this layer only, we have lots of chloroplast, and where the photosynthesis takes place. So this has so many chloroplast on it. Okay, it has so many chloroplast on it, and see, the basic thing is it is tightly packed with each other and has no intercellular space. That we need to keep it in our mind. Okay, it has no intercellular space. Okay, after this we have one more layer which is called the spongy layer. Okay, second layer which is called the spongy layer. Okay, now let's see. Now we have the lower epidermal. Okay. This way there is lower epidermal, and you see there is a gapping in between the lower epidermal, which is stomata, and in this stomata there are guard cells which is of bean shaped. Okay, which is of bean shaped. Okay, so this is how the cross section of leaf. And what is this section? Let me make you understand. Let's suppose this is a leaf. I have cut this leaf in this way. Okay, horizontally I have cut in this way. Now I have two parts. Let us suppose this part done, or now this is this way, the cut plant, rest of the part of leaf is there. Now I will make the leaf like this, and this section is this section. Okay, this section has been drawn in this way, right? Now let's understand the what are the, let's label it, but before this let me make it one thing here. Okay, now let's see what are these things. First of all, this is upper epidermal cell. Okay, before this we have cuticle. Okay, then we have column mesophil. Okay, column mesophil. Then this one is called spongy layer, spongy mesophil. Okay,
In between this, you see the thing which is in between is called intercellular space. Intercellular space. Now you see the gapping. This gapping is called stomata. Stomata. And this blue color thing is called guard cell. Guard cell. I hope this is clear. And this green thing which is here also, in this also there are so many green things, not only one but many. So these are called chloroplasts. These are nothing but the chloroplast. These are nothing but the chloroplast. Okay, I hope it is clear. Now let's see why am I telling you this figure. Why is it very important? Because to understand the transpiration and its kinds, its types. Okay, so first, transpiration has been taken place through the cuticles. What are the cuticle? Cuticle is a waxy layer. It is a waxy layer on the surface of a leaf. Okay, every leaf has a waxy layer, maybe a little less or more. Okay, but maybe sometimes it is a very thick layer and sometimes it is a very thin layer. So when you see, it's not even all the sides, right? Somewhere it is very thin, somewhere it is very thick. When the layer, this cuticle layer is very thick, then transpiration will not be able to take place. But when there are such areas where this layer is little thin, then the transpiration will take place through this way. Okay, but what is the function? You'll be very surprised to know that function of the cuticle is cuticle prevents the water loss of the plant. So basically this cuticle has no function like transpiration, and not in fact any part of the plant has special function for transpiration, because plant is not designed or not ready for the transpiration, because they don't want that how having lots of effort they put to get the water, and once they absorb the water they don't want to lose it. But there are some circumstances where they need to do this, and this is one of the very important process they have to do, but they are not ready for that. That is why they don't have any developed part so specifically for that. Okay, so that is why when we will understand the function of these things like cuticle or lenticle or stomata, they don't have the function of losing water. Okay, transpiration is not the function of any of these aerial parts, right? So basically the function of cuticle is, as its structure is very waxy, so it doesn't hold the water. That is why its function is to prevent the water loss. But obviously ironically it is doing transpiration, because, and yeah, that you need to understand, thick layer of cuticle will prevent the transpiration. Transpiration will take place only and only when the layer of cuticle is very thin. Okay, let's understand the lenticular transpiration. It takes place in the lenticles. Lenticles is where do we see the lenticles? Bark of trees. When we see the bark of trees, that the brown part of the tree, that that is the lenticles present on that. See, lenticles are the obviously pores. Lenticles are the pores. But how is it different from the stomata? Stomata has closing and opening, right? Stomata is not open all the time. That is why we can't do, means the photosynthesis also not been done every time. So pores, but lenticles are always open. They don't close. Okay, they don't get closed. It is always open because it is on the surface of the plant. What is the function? What is the function of the lenticles? It plays major role in gaseous exchange. Okay, in plant also we know that gaseous exchange take place. So lenticles are for that only. They let the oxygen be out and carbon dioxide let in, because we know that in plant also respiration takes place. So obviously gaseous exchange is going on. So for that lenticle works, it plays a role there. But it again, it doesn't have the function of transpiration, but it takes place since there is a pore, there is a pathway, there is a gateway through which water vapor gets out. Okay, so this is the two types of transpiration. Now let's understand the maze. This see, this is very important. Okay, this is very important, keep it in your mind. So now let's see the third part, third kind of transpiration. Third kind of transpiration is stomatal transpiration. Okay, again the same thing, stomatal transpiration takes place through the stomata. Stomata are the tiny pores through which gaseous exchange take place in plants. It is present especially on the surface of leaves. See, it is especially present on the surface of leaves, but also present on stem as well. Okay, so stomata are the tiny pores. So from this pore only, when the water vapor gets out and transpiration takes place, this type of transpiration is called stomatal transpiration. So let's understand the diagram of stomata. See, there are two types of stomata. One is the open stomata and second is the closed. So we will understand the open one first of all. See, stomata are the kidney shaped. Okay, what is the shape? This is how the stomata looks, but it is not it. We have to draw some of the more arrangements. Okay, let us suppose what are the arrangements we are going to do. Let me erase this part so that we can get the place. Okay, what are the space? This is the epidermal part. We know that. Okay, this is the epidermal part of a leaf, of the stomata. Okay, now again this stomata is a living cell, so it has nucleus. It has nucleus. It is the part of plant so that it has vacuole, very big size vacuole. Okay, I hope it is clear. Now obviously it has lots of chloroplast in it. Okay, it has a lots of chloroplast in it. I hope this is clear. Let's level it. When you level it, you'll find out the first part is the epidermal cell. Okay, the first part is the epidermal cell. Okay, now this opening is called the stoma. Why stoma? Because it is a single unit. One stoma is only there, that is why it is called a stoma. Next is this is called the nucleus. This is called the nucleus. Okay, next is this one, it is called the vacuole, food vacuole or the vacuole. I hope it is getting clear. Now the green parts are called the chloroplasts. Okay, this is the chloroplast. Also, this is the cell wall. In it, there is a cell wall also. So, this is the way how does a stomata look like, and how can we draw a stoma in this way. And which type of stoma is this? This is the open stoma. This is the open stoma. I hope it is clear. This you need to understand and keep it in your mind that this type of diagrams can be can play a very important role in your examination. So don't skip these diagrams. And you must have seen the drawing diagrams are so easy, so easy, right? So you can also draw it, and if you are drawing together with me, it will be more beneficial for you. I hope this is clear. So stomatal transpiration means the transpiration taken place through the stomata of a plant, and stomata are present especially in the leaves and the stems. That is the aerial plant, on the aerial plant of a tree or a plant. Okay, I hope it is very clear to you. Now let's do the next topic. So this much is clear. Let's do the next topic. This is also one of the very important topic. That topic is factors affecting the transpiration. That means those things which can increase or decrease the rate of transpiration. So first thing is about the sunlight. Okay, see, sunlight when will be more if the intensity of the sunlight is more than the rate of transpiration will also be more. Okay, when the intensity of sunlight will be high, means when there is lots of sunlight, then the transpiration also will be more. If the temperature is more temperature or high temperature will increase the transpiration. Okay, what happens? More temperature, more transpiration. More sunlight, more transpiration. That means these are the directly proportional thing, right? What is directly proportional? When one thing is high, second thing is also getting high. When one thing is getting high and second thing is low, that is called inversely proportional. So this is again increase in the velocity of wind will increase the velocity of wind will increase the transpiration. That is the rate of transpiration. Okay, so if the velocity of wind is very high, means wind is blowing in a very high velocity, then the transpiration also will be more and more. So the rate of transpiration will increase. Now the humidity, from here all the things are indirectly proportional, like inversely proportional, indirectly, not inversely proportional. So increase in humidity will decrease the rate of transpiration. See, simple thing is that transpiration is the loss of water vapor, means water is getting converted into the form of vapor, and it happens only and only when the temperature is high, not in the low temperature. And humidity means when the air has lots of moisture. When the air has moisture, obviously its temperature will be lower down. So that is why the transpiration will also get decreased. The rate of transpiration will be less. See, again about the carbon dioxide, increase in the level of CO2 will decrease the transpiration. Okay, with this will decrease the transpiration. Now atmospheric pressure also has the same thing. increase in the atmosphere pressure will decrease the transpiration. Okay, I hope it is clear now that rate of like temperature increase in the atmospheric pressure will decrease the rate of transpiration. I hope this is clear. So these are the external factors which affect the transpiration or the rate of transpiration. I hope this is very clear to you. So just you need to remember that after humidity all the factors are like inversely proportional. Okay, now let's understand a very important topic, which is guttation and bleeding. See what happens, as we have discussed now, when the humidity is more, the transpiration will be less. Okay, so what is happening, the plant and its roots is absorbing water continuously, right? It's absorbing the water continuously and this water is getting up. So what happens, the transpiration takes place, then water goes out. But what if the transpiration doesn't take place? When the case of, in the condition of humidity, okay, in case of humid condition, okay, in the case of humid condition, obviously transpiration doesn't take place. When the transpiration doesn't take place, then that water which has been absorbed by the root goes to the margin of the leaf. Okay, let's suppose this is a leaf and the margin of the leaf has this tiny droplets collected. Okay, now in this margin there is a cell called hydathodes. Okay, hydathodes. These cells are present. Okay, so these cells are present and here the this type of tiny droplets you must have seen during early morning. You must have seen on the leaf there are some tiny droplets of water. Okay, especially in strawberry plant or banana plants you must have seen this. This is what this is called guttation. See, guttation has the meaning to pour out, to pour out. Okay, this is why, because the transpiration is not taking place, so that water obviously will go somewhere, so it will be get collected on the leaf, margin of the leaf, and this is called guttation. Bleeding is in the case of injury in plants. Again, this is a very surprising thing to know about, that plants also get injured, and when it get injured it bleeds. Okay, so that is that water which comes out or secretes out during the injury of the plant is called bleeding. That is why we consider plant as a very close living being, and we have lots of experiment that to prove the similarity between we and plant. Okay, because plant also have many of the things which they do like us, like bleeding. Bleeding is one of them, right? As we get injured we bleed, the same way plant also gets bleed. So this is what guttation and bleeding is all about. So let's understand some of the, okay, here one thing I'll be talking about, that is called the suction force. Suction force or the transpirational pull. Okay, as I told you about the suction force, that the plant when have the void created, the plant root sucks the water to put it upward to fill that void place, that empty place, and this force, and through this force, this is called suction force. Here one thing also takes place, that is active transport of water. What happens, the water and mineral, that sap, it uses its energy to suck or to bring the water, okay, that bring the mineral to it. Once the mineral is there, there is water concentration high. There is high water concentration and there is mineral. So it has low concentration of water. Okay, here water concentration is high, here low. So we know that the rule of the osmosis, the water will goes in, and this is called the absorption. This is the way how they absorb. So this is the active transport through which they absorb the water and mineral through it. And suction force is the force when there is a void created by the water. So next of the waters or rest of the waters goes or runs through to fill that gap, and that is called the suction force. So this is 1-2 things which we needed to understand. So we have understood it well. Now let's understand some of the important differences. Important difference between evaporation and transpiration. Let's understand evaporation. Okay, evaporation and transpiration. Evaporation and transpiration. See, evaporation takes place on the surface of a body. Okay, or here transpiration takes place through the aerial parts of a plant. Okay, this is a complete physical process and gets affected by only the external factors like humidity. Okay, like humidity. Now this is a partially physical process and gets affected by both the external and internal factors. I hope it is clear. Again, evaporation is a fast process and transpiration is a slow process. I hope these differences are very clear to you. First difference is evaporation takes place on the surface of the plant, and it takes place through the aerial part, like the leaves and stem of the plants. And it is not about plants, it's the body, any surface it can be. Okay, next is it is a complete physical process and also been affected by some external factors like humidity, weather condition. Okay, again evaporation is a very fast process. This is partially physical, this is not completely physical process, it's a biophysical process although. So this is not a completely physical process and gets affected in by the both of the conditions, like external factors also can affect it or the internal factors. And transpiration is a very slow process. I hope these differences are very clear to you. Let's understand the next difference, which is the difference between transpiration versus guttation. Okay, transpiration and guttation. Okay, see what happens in transpiration. Transpiration has water loss in the form of vapor. Guttation has water loss in the form of water droplets. Okay, first difference is that only. Second difference is transpiration transpiration takes place through cuticles, lenticles and stomata. Okay, and guttation takes place through hinder thoughts only. Okay, hinder thoughts only. Next is third part is stomatal opening and closing can be controlled by the guard cell. Okay, but the opening of hinder thoughts can't be controlled by any cell. Okay, hinder thoughts can't be controlled by any cell. Stomatal opening can be controlled by the guard cells. This we know. Okay, transpiration as water loss, and in this water doesn't only get lost in guttation but also the minerals. Okay, that means we can say the sap, but here only loss of water takes place. I hope these differences are also very clear. Significance of okay, one thing more we will be discuss orally about, and that is how can we prevent the transpiration, because not all the plants are fortunate enough to afford this transpiration, such as like xenophytes, those plants which grow in the desert areas. How will they survive with this transpiration? So obviously they don't have the potential to bear with this transpiration. So what can they do? They can have sunken their stomata, means if the pores, okay, that pores through which the transpiration is taking place, if that can be minimized, so we can obviously prevent the transpiration. You must have seen the cactus or this type of like plants which grows in the desert area. They don't have actual leaf, but they have the spawn like spores like structures, right? What are they? They are the sunken stomata, right? They have been shrunken stomata, that is why so that the less pore means less transpiration. Number of pores will be more, number of transpiration will be more. That means the number of stomata is high, transpiration rate of transpiration will also be high. So they don't have that very wide leaf, they have very narrow leaf. They have spores instead of the leaves. Okay, so these things are there, so they survive in this way. Next thing is about the significance of the transpiration. Transpiration has first importance, that is the creating suctional force. As I told you in before, suctional force is the one of the very major force which is very essential for the absorption of water and mineral in plants. So this transpiration creates this suction force through which the water gets absorbed. So that means it is one of the very important and essential process of plant, right? Next significance is the cooling effect of not only for plant, also for the environment. So this transpiration needs to be done due to that, because this of the transpiration, the environment also gets cooled and the plant also, the temperature of plants also gets regulated. So this is why these are the significance of the transpiration. That transpiration, why is transpiration important? If this is the question, then this can be the answer. I hope you understood all these things very clearly. So if you have enjoyed the class, do like our video and also share it with your friends so that they also get benefited. Subscribe to our YouTube channel Midha Guru, and also this type of amazing contents if you want in Instagram, we have our Insta ID. Follow us there also. There also you will surprised to see so many amazing contents. Also, if you have any doubt left through about this chapter or any of the topic of biology, do comment down below, and the comment section is open for you. So do comment down below and do let us know if you have any doubt. I'll make sure to make your doubt clear as soon as possible. So let's meet with the new video, new amazing class. Till then, bye bye and take care.